Thank you for joining us this evening for Lupus Canada's Let's Talk Lupus Disease Awareness webinar. Tonight, Dr. Hiraki will be discussing the genetics of lupus, what we know, and where we're going. Dr. Hiraki is a clinician scientist in the Division of Rheumatology and scientist in Genetics and Genome Biology program at Sick Kids Research Institute. Dr. Hiraki is coordinating a large international cohort of systemic lupus erythematosus patients followed prospectively over the years of disease to understand the genetics of lupus. That is a mouthful. It only manifestations, the disease course, and related outcomes. She is also leading studies to identify rare genetic changes leading to systemic inflammatory diseases, including SLE and neonatal lupus erythematosus NLE studies. Just a few housekeepings to address before we begin this evening. Everybody will be placed in listen-only mode, therefore your audio and video will be disabled. If you have a question, please put it in the chat box and we will be sure to address that at the end of the webinar. We hope you find this webinar informative. I will now pass this over to Ariana Ranjabar, who is our moderator for this evening. Ariana?
Thank you, Leanne. So I'll be the moderator for tonight. So if you have any questions throughout the webinar, please put it in the chat box. And if it's urgent, I will address it right away. If not, then we'll wait till the Q&A at the end, and I will make sure to ask Dr. Hiraki at the very end. So now I'll pass it off. Thank you for joining us tonight.
Thank you. Thank you, Ariana. Thank you, Leanne. Thank you, Lupus Canada, for the invitation. And thanks to everyone in the audience for being here. I hope to offer a little bit of knowledge. There will be a couple of interactive questions. So let's sort of dive into it. But I do have a disclaimer to begin with. The information that I'm going to talk about is for educational purposes, and it's really designed for this audience. So I ask you kindly, please don't take screenshots or copy it. If you really would like to get more information, please feel free to contact me directly, and I'm happy to share my email contact. Okay, so with that, let's go into it. Systemic lupus erythematosus, SLE or lupus, as I'm sure everyone here knows, is a very heterogeneous disease. So what do we mean when we say that? We mean it looks different in everybody. It's been called the disease of 1,000 faces precisely for that reason. It can affect virtually any organ system, and no two patients are exactly alike. It's a chronic disease. Unfortunately, today there is no cure, and it means that there are periods of time where the disease is active and more quiescent. It's an autoimmune disease. In fact, some would call it the prototypic autoimmune disease. And when we think of the immune system, as I often describe it to our patients and families, is the immune system is designed to protect us from those infections like COVID. And in some instances, that immune system can start attacking our own tissues. And that is definitely what's happening in lupus. It is also life-threatening. It is a serious disease with serious associated mortality risks and morbidity risks. Thankfully, with increasing therapeutic options, the life expectancy has increased a great deal for lupus in recent years. So when we look at the epidemiology, or what this disease looks like in populations, we know that the prevalence of lupus is about 50 and 100,000 in Canada, but the range can be very different depending on the population you're looking at and the country and the ancestry of a population. And I want to make the distinction between ancestry, which is what part of the world your great grandparents come from, versus ethnicity, which often refers to how a person identifies, how they see themselves. In this figure on the right, it's taken from a paper that we published on our lupus clinic population at Sick Kids. And we wanted to look at the demographic of the clinic population compared to the Greater Toronto population. And what you see on the x-axis are different ethnic groups, and this is based on self-report, how people identified, and the proportion they comprised within the pediatric lupus clinic population in black and the Metropolitan Toronto at-risk population in gray. And what you see straight away is that there's a larger proportion of patients of Asian, Black, South Asian, and mixed ancestry in the Sick Kids Lupus Clinic cohort compared to the general at-risk Metro Toronto population, which tend to have a higher proportion of Caucasian people who identified as Caucasian. And this is certainly true of lupus in many different regions of the world, that generally it's a disease that is more prevalent among non-European populations compared to European populations. We also know there's a strong female predominance. In fact, the ratio of females to males in adults is 9-to-1, and in children 4-to-1. And speaking of children, about 20% of people who are diagnosed with lupus are diagnosed under the age of 18. So I now have my first question for you. What factors have been identified to increase the risk of developing lupus? A, genetics, B, infection, C, ultraviolet or UV exposure, D, smoking, or E, all of the above. And Ariana is going to be looking at the chat. So if you guys want to put in your answers in the chat, she can take a look at what the most popular ones are.
So far, everyone is saying E.
Oh my gosh, this is a highly informed audience. And I agree. That is it. Each and every one of these factors have been implicated to contribute to the risk of developing lupus. And I think that's because what we know so far is lupus, like many diseases or even phenotypes, are a result of both environmental factors interacting with genetics. So on the left, you have a couple of those environmental factors that have been linked as contributors to lupus risk. And on the right, I'm listing some of the epidemiologic evidence we have that implicates genetics as being important. I already talked about the female predominance, the fact that it tends to be more common in non-European populations. And then another piece of information is that when they look at identical twins, so twins that are genetically identical, and compare their concordance, so the number that both have lupus, to fraternal twins, which are genetically more like siblings, there's a much higher concordance rate. So siblings or identical twins are more likely to both have lupus than if you were to look at fraternal twins. So all these things indicate that genetics is playing an important role in determining who will get lupus. And when we talk about genetics, what are we talking about? We're talking about the study of heredity, that passing down of traits or phenotypes and how they're inherited from generation to generation. And genetics can be simply thought of as perhaps these Mendelian or monogenic traits. And these are those diseases where there's a change in a single gene that then leads to disease. And this is a figure that I borrowed from the NIH website, where they show a pedigree or a family for Huntington's disease. And here on the bottom, you see that males are in boxes and females are in circles. And those that are shaded in dark blue are affected, so they have Huntington's disease. And those that are unshaded are unaffected, they don't have it. And this is an example of a disease where all you need is one change in a gene to result in disease, compared to a disease like sickle cell anemia, where what you need are actually two copies to get the disease. And here, the carriers, people who carry just one copy, are in a dot. And you'll see that if you go back 3 generations, there were carriers that mated with other carriers, and that resulted in an affected child, where there was 1 that was unaffected and 2 that were carriers. So these are the general concepts we think of when we think of these Mendelian or monogenic traits. And we can compare that to polygenic traits. And this often is the case for very common diseases like coronary artery disease, where really what we know is it's not a single genetic change, but rather hundreds and hundreds of changes across the genome. And each of these 2 lines represent different chromosomes. And these are the sex chromosomes. And you can see here, the idea is people can carry 2 copies at the same site, or they can carry 1 copy at a site, but it's all of these things together that we believe are contributing to the risk of disease. And that's what we think is going on for diseases like lupus. And so if I take you back to your biology 101 and you think about the genome and what's part of those chromosomes, it comes down to the DNA, deoxyribonucleic acid. And we know that we have 2 of each of the chromosomes that at one given site, you may have this type of nucleic acid, or you may have another one. And when people talk about single nucleotide polymorphisms or SNPs, this is what they're talking about, the variation, the polymorphisms between individuals. And in this instance, this person would be considered having one copy of A and one copy of C, but another person may have two copies of A and two copies of C at the same site. And this is what people are studying when they're looking at large populations and trying to link these changes with different traits. So again, when you think of how our genome can be exerting its effect, these changes in the DNA, which are like instruction manuals, could result in changes in proteins. However, proteins, or the exome, comprises only a small proportion of the entire genome that's made of 3 billion base pairs. The exome is only about 1% of that. So that means the rest of the gene, what is it doing? Most of our DNA is probably involved in regulating not the structure of the protein, but how much of it is there, whether there's a lot or too little. So why study genetics? I think there's a host of reasons, but I'm also very biased. I think in some instances, we want to quantify the proportion of risk that's inherited, as well, conversely, to understand how much is influenced by the environment. Knowing about genetics can provide deeper insights into the biology of disease. The hope is maybe it can help improve prediction, as well as design and implement targeted therapies. And ultimately, I think all of these are building towards the same common goal for disease prevention and ultimately cure. So this is a table that's very simplified, but just to sort of conceive of heritability and think of it in relation to other phenotypes. And when people talk about heritability, they're often referring to the proportion of risk or the proportion of variation in a given trait that's attributed to genetics versus all other things. So here you can see things like type 1 diabetes and psoriatic arthritis and height are considered highly heritable. Genetics plays a large role in determining how tall you will be. And here is lupus, and you can see it's definitely on the higher side, not quite as high as type 1 diabetes, but more so than rheumatoid arthritis, osteoarthritis, or juvenile idiopathic arthritis. And oftentimes when people think about the relationship between these SNPs, polymorphisms or variants, they think about how frequent or how common it is in a population. And usually for polygenic phenotypes like coronary artery disease, where you have multiple changes in genes across the genome, these variants tend to be relatively common, present in 5% or more of the population. And individually, their effects are usually small. So it would take, like I said, hundreds of these changes to result in the phenotype that you see. And so to study these, we often need these arrays, and we often need tens or hundreds of thousands of people to be able to pick up these changes that are quite common. And so on this graph where you have the allele frequency, so over here are things that are more common. On the other side, you have things that are much more rare. These rare alleles that cause those Mendelian or monogenic diseases that we were talking about before, like sickle cell or Huntington's. And these single genes changes are very rare, and they usually have very large effects. And the way we identify these rare changes is by using very specific sequencing, so identifying every nucleotide across the genome. And so these days, those are referred to whole exome or whole genome sequencing or Sanger sequencing. So you're getting really, really detailed information on every individual. So the next question. For most people with lupus, and I'd ask you to complete the sentence. A, genetics are not risk factors. B, a change in one gene leads to lupus. C, changes in 10 genes increases the risk of lupus. Or D, changes in over 100 genes increases the risk of lupus.
We have a B.
Okay.
Oh, we have a C.
Right.
Another B.
So I'm going to say the key here is for most people. That's the question, for most people.
A few people said D as well.
All right.
Mix of different answers.
Wow. Okay. Well, so the answer, I think, is actually D, that it's changes in hundreds of genes. And hopefully I'll show you some evidence to convince others that this is indeed the case. So genome-wide association studies have been used commonly to study tons of traits and diseases. And what they're doing is they're genotyping thousands and thousands of people and then saying, okay, I don't know what genetic changes will lead to height or coronary artery disease or lupus, but I'm going to see what I get. And there have been a number of genome-wide association studies, or GWAS, for lupus. And this is one of the larger ones that was published in 2017, where they took about 11,500 patients with lupus and nearly 16,000 non-lupus controls. And what they were able to do is identify, sorry, this is a Manhattan plot. It's called that because it's supposed to look like the Manhattan skyline. Some creative thinking. So on the x-axis, every chromosome is a different color, and the y shows you how strongly each of these dots or SNPs are associated with lupus. This little gray line at the bottom represents what we would consider significant, what's strongly associated. And as you can see, there are many, many, many dots that are exceeding this line. And some of these are very strong, like HLA, which is human leukocyte antigen. And it's one of the most variable regions in the entire genome, and it's involved in our immune responses. So it's actually not at all surprising that it's one of the strongest risk factors for lupus. But each of these little dots are actually a risk factor. So in this particular study, they identified over 90 independent SNPs that were associated with lupus. And what I'm not showing you is the subsequent study a couple of years later identified over 120 SNPs that were independently associated with lupus risk. So it's studies like this that tell us that for most people with lupus, it's actually a consequence of probably over 100 changes across the genome that's leading to disease. But those who answered B are not totally incorrect, and later on in the talk, I'll tell you why. What's interesting about these GWAS loci is that they all tend to be in or near genes that are involved in immune responses. And this is a very busy cartoon, but just to show you that our immune system is involved in many different activities. Cell death, which is part of the normal process of life, clearing some of those cell fragments and generating antibodies. And all those peaks can find a map to one of these genes involved in innate responses and adaptive immune responses. So what have we learned from GWAS of lupus? Lupus is a polygenic disease. It has roles in innate and adaptive immune genes. Many loci lie in those non-coding regions I was talking about. So we think that means that the role they play in lupus risk has to do more with how much or little protein is expressed. However, there's lots of unanswered questions that we still don't know. Why is there this female predominance? What determines when a person will get lupus? Why is there such differences between what lupus looks like? Why do some people get kidney disease and others don't? How can we use this information to understand disease course, when people will flare, and who will respond to therapy? So we've learned a great deal from genetics, and there's still much that we need to know. Okay, so this I think is my last question. There is evidence that genetics influences the age of lupus onset. So this is a true or false?
Give everyone a few seconds. We have a true. True. More truths.
Okay, so I don't know, maybe it's a leading question. Yes, it's true. It's absolutely true. And let me show you some evidence why. So first off, if we reflect on what the disease looks like in children versus adults, there are studies that have shown that perhaps the disease tends to be more severe. There's a greater prevalence of people with lupus nephritis, kidney disease, a larger proportion of children compared to adults who have neuropsychiatric lupus, so brain involvement. There also tends to be more hematologic disease, so low blood counts in children compared to adults. And it's been suggested that maybe that reflects a higher genetic burden for disease. And a couple of studies have looked at the relationship between the number of these common risk alleles, these changes in the genome, and how it relates to the age of diagnosis. And one of the earlier studies to do this was published in 2011. And here on the x-axis, they've plotted the age of lupus onset. And on the y, they counted up how many lupus risk alleles a person had. And the higher it was, the younger their age of diagnosis. However, this was significant only in those of Gullah or African American ancestry, but less so for those of Hispanic or European American ancestry. So again, this is this hint that relationship is not uniform across all ancestral groups. Interestingly, we wanted to look at this within the population that we've genotyped, and we genotyped nearly 1,000 children and adults of European ancestry. And these are patients from across the world. And what you see is there's this negative relationship. The higher their genetic risk score, and this is, sorry, it's using a bit of code here, but it's a genetic risk score where we took all the alleles that were associated with getting lupus, and we added them up, but we also gave them a weight, so we let them contribute different amount of risk. And the higher a person's score, it seemed like the younger they were at diagnosis. So again, multiple groups have replicated this phenomenon. But what I'm actually not showing you is what the score was for this youngest group, and acknowledging that they are quite young, under the age of 5 when they were diagnosed, and it's a small group. But interestingly, their score is not the highest. You might expect it's all the way up here because they are so young. But in fact, it was a little bit lower. And this makes us wonder, well, why is it that these very young children are getting lupus when they don't seem to have the same burden of common lupus risk variants? And what we suspect is that they represent not that polygenic disease, but instead the monogenic lupus. And this is where those people who answered B were correct. But my point is that I think for the majority of people, in fact, there's evidence that for the majority of people, lupus is a polygenic disease, that it's changes in many, many genes that results in disease. But a much smaller proportion of people, it's a monogenic disease. And I think one question I'd like to pose to the group, and I think that a lot of people are thinking about, is whether studying monogenic forms of lupus can teach us about the polygenic forms. This notion of monogenic lupus is not new. It's been around for decades. In fact, the earliest recognized forms of monogenic lupus were in complement deficiencies. And with the onset of genome-wide sequencing, next generation sequencing, where we're able to sequence more and more people and really zoom in on those specific changes across the genome, we're learning and identifying more and more genes that we believe are involved in monogenic forms of lupus. And they're involved in a lot of those pathways that I was showing you before in that busy cartoon: complement or immune clearance, cell death, DNA sensing, and inflammatory pathways. And to illustrate this point, I'd like to talk about patients that we cared for in the lupus clinic. It's the kids. They were sisters who we met, of Turkish ancestry, born to parents who were first cousins. When we met one of the sisters, she came with a diagnosis of lupus at the age of 2. Her disease was comprised of joint pain and swelling, so arthritis. She had low complement levels, a very high anti-nuclear antibody ANA titer. But she also had a couple of features that were not typical for what we would see in many of our other lupus patients. She would get these fevers, and when she'd get them, she'd get swelling of her lips and her face. She'd get hives and red eyes. And when we saw her, she was on Plaquenil. Now, this was sorry, and if I were to show you what her rash looked like, it looked like these little hives. This is not our patient, but this is what she looked like. And I should mention, we have consent from the family to talk about them. Her sister was diagnosed with lupus at the age of 5. She also had fevers and arthritis, but she also had kidney disease. And when we met her, unfortunately, she was dialysis dependent. Now, what was so striking about this family is that the girls were extremely young when they were diagnosed, but also there were some unusual features in the sister who was diagnosed at 2. And this is a pedigree of the family where you can see the parents are first cousins. And both of these girls were diagnosed at young ages. And there was also a history of autoimmune disease on mom's side with a cousin also diagnosed with lupus. So it was this strong family history and the fact that one of the sisters had these rashes, these hives and fevers, that we wondered whether there could be something monogenic. And we sent genotyping, and sure enough, the sisters were homozygous, meaning they had 2 copies for a deletion in their gene, this gene DNASE1L3. And this same deletion had been reported in a couple of other families in the literature, also of Turkish ancestry. So it predicts basically that the protein DNASE1L3 is short, and that means it gets degraded and it's non-functional. So what does DNASE1L3 do? Why does it matter? So under normal circumstances, DNASE1L3 is an enzyme that goes around kind of cleaning up fragments of DNA in the circulation. So as I mentioned earlier, a normal part of our body is our cells are constantly turning over, and that means we get these little broken up fragments of DNA. And the DNAse clears it out of the circulation so that our body doesn't see it as something foreign that needs to be attacked. And that means we don't make antibodies, these proteins, against DNA. So in the absence of DNASE1L3, you actually get persisting fragments of DNA that get detected by our cells, and then they can start generating these antibodies, these anti-double-stranded DNA antibodies. So there was an interesting paper that was published in 2016, and it was a small paper, but they looked at 20 lupus patients and 26 healthy controls. And these are people who have presumably polygenic lupus, right? They don't carry these very, very rare changes in DNASE1L3. But what they found is in lupus patients, there was a lower level of DNASE1L3 enzyme compared to healthy people, and the activity of the enzyme was also lower. And then again, recently, there was a study of 50 controls, so 50 healthy individuals, 4 people who had a DNASE1L3 homozygous deletion, 85 renal lupus patients and 35 non-renal lupus patients, and 45 anti-Ro antibody patients. And what they observed was that people with lupus with renal disease had much lower levels of DNASE1L3 activity compared to healthy controls. Not as low as people who had the deletion, like the sisters I showed you, but much lower and significantly lower. They also showed that these people had higher levels of anti-DNASE1L3 antibodies, which they demonstrated in this paper was neutralizing. So these antibodies actually made it so that the DNASE1L3 couldn't perform its action. So this was a really beautiful example of how a rare process gave us clues to a disease process in a much larger number of people with lupus. So some of the take-homes here, DNASE1L3 rare variants have been linked with monogenic lupus, and knowing this information can help with management and prognosis. That there is a role, it seems, for DNASE1L3 in polygenic lupus, and that most recent paper gave us some insights into pathogenesis. So I think what we're trying to do in our lab, our small contribution, working with many others from around the world, is to try to identify more cases of rare variants, but also by genotyping many people with lupus, trying to make that link to polygenic lupus as well, and try to understand the interplay between those rare and common genetic changes and how they lead to lupus. So with that, I hope I've given you an idea that genetics play an important role in lupus, that they can provide these insights into pathogenesis, that hopefully it can help us better screen and provide better treatment for patients and families, and that we can learn different things from studying these rare variants as well as the common variants. I want to acknowledge all the many people I get to collaborate with, the funders for the work I shared, and most importantly, I want to acknowledge and thank the patients and families that I care for and who agreed to participate in this research. And thank you all for your attention. I'm happy to take any questions, and I can stop sharing so we can talk.
Thank you so much for the presentation.
I hope I wasn't too long-winded.
Absolutely not. Someone has already said thank you for the presentation.
Pleasure, Heidi.
Oh,
Where am I from? I work at the Hospital for Sick Children in Toronto, and I'm from Toronto.
A lot of praise so far.
Great, thank you. I wanted to make sure that it could meet everyone where they're at. So I'm sorry if it was too basic for some and too complex for others. I was trying to strike a balance there.
I think it was an excellent presentation, and it's a lot. I think a lot of people are probably going to digest the information that you provided. We will also be able to share this presentation on our website within about 48 hours. So if folks want to look back on it and go over some of the stuff that was presented this evening, it will be available.
We do have a question. Do you think this genetic analysis can lead to prevention strategies?
You know, I sincerely hope so. So there's been some interesting initiatives that have been that have taken place in many, many diseases. And I think, you know, when we reflect on lupus and rheumatology, we have a lot to learn from other diseases that have been studied for many, many more years. But I think this notion of risk and whether we can predict who will evolve to develop lupus, to my mind, that's one of the key rules for prevention. So if we believe there are certain people who may be genetically more susceptible, is there a way for us to intervene either with a medication early on before they develop full-blown disease? And I think there have been some interesting studies. In fact, the trial that was looking at early lupus and using Plaquenil or hydroxychloroquine, which I'm sure everyone here is familiar with, but to see whether if you had a population of people who didn't really have severe features of lupus, maybe they had a positive ANA, maybe they had a malar rash, or maybe they had a very strong family history, and you were to say, I'm going to give one group hydroxychloroquine and another group a placebo, and see whether it will delay progression of disease. That study is actually, I think, concluding recruitment and going into analysis, but there are older studies in Army recruits that give us a clue that Plaquenil may delay progression of disease. And so I think one of the challenges I think we face as clinicians is trying to know who those people are. And if there were ways for us to identify them early and to say, okay, now these are the people I should follow and potentially have a low threshold to start hydroxychloroquine because I think it's going to make a difference to you in the future. I think that's one very simple strategy that can make a huge difference for individuals.
Thank you for that. We have a few questions, but feel free if they're a little bit too personal, perhaps they can be redirected after the webinar to either Leanne or yourself. But someone has asked if developing lupus at the age of 72, is that kind of old to develop lupus, I guess? That's the question.
So I hear what you're saying. I think certainly when you read the literature, it's typically thought of as a disease of young women, right? But I mean, it doesn't mean it can't ever happen. I just gave you an example of two very, very young girls who were diagnosed at 2 and 5. And I think the same is true across the age spectrum. I think, unfortunately, this speaks to our challenges in being able to really understand fully the disease. So unfortunately, I think I'm glad that it wasn't until you were 72. And I don't think that the fact that it was at 72 means it's not lupus. I don't know if that was part of the question, but I think it certainly can happen. There's no question about it.
Thank you. Someone has asked, so how likely is it that someone with likely polygenic lupus can pass lupus on to their children? Asking as a mom of 2.
It's a great question. It's a question I get a lot in the neonatal lupus clinic, where I meet a lot of moms who have rheumatic disease diagnoses. So what we know from epidemiology is if you have a first-degree relative, so that means a parent or a sibling who has lupus, your individual risk is much greater than the general population who does not have a first-degree relative with lupus. Quantifying it is very, very challenging because we don't know specifically what the risk is for each individual. There have been groups that have tried to individualize risk from using these large studies. I'm not convinced that they've been successful. I think this is where it's a hard balance, because you don't want to give someone a diagnosis if they don't have it and they don't need it. At the same time, you want to be vigilant. So I think what I often say to people is, until you have signs or symptoms, there really is no reason to think that a person has it. It's not like those monogenic diseases where you know very confidently if you have 2 copies, you have the disease. The polygenic one is much more complex. And I think the other piece I will say is we also have to acknowledge that it's not like a fatalistic process here. Environment also matters a great deal. Some of those things you could control, like UV exposure, other things you can't, like a global pandemic. So I think it's a tough balance. I don't know if that was a very satisfactory answer, but I hope it helps.
I think it was great. She said, thank you. So someone asked, how do you diagnose lupus nephritis?
So I think this, there's a really, the way you do it is you look at a person's blood work and their urine and their physical exam. The only way you can ever know what type of lupus nephritis a person has, because there are different types of kidney disease and all under the umbrella of lupus, is with tissue. You have to look at it under the microscope. But you can certainly have an idea that someone has inflammation in the kidney when you look at their urine and their blood work and do a physical exam.
Thank you. Don't have any more questions as of now. But I don't know, Leanne, if you wanted to add anything.
Yeah, really informative. I always learn so much from all of these webinars. So thank you so much. And tonight's was definitely a good example of somewhere for us to continue to learn about lupus, about the genetics, and very, very informative. So we really appreciate you taking the time, Dr. Hiraki, and presenting this to all of us. I know, like most, I'm going to revisit this webinar and continue to learn. So we really appreciate that. I think there was one more question that just popped up, so we don't want to miss that. Ariana, do you want to address that one?
Yeah, someone has asked, is there a way people with lupus can volunteer to donate their DNA to these kinds of studies?
Yeah, absolutely. I mean, I think someone else had commented very clear, like Dr. Urowitz, which is a huge compliment. Oh my goodness. Thank you so much. So I actually collaborate with Dr. Urowitz and Dr. Gladman and Dr. Tuma at the Western, as well as many other doctors who care for lupus patients across Canada. And I would say, if you are interested, by all means, please speak to your rheumatologist. And if you want to reach out to me directly, I'm very happy for you to do that. I'm happy happy for Leanne and Ariana to provide my email contact, and we can try to figure out a way, because I agree. I think it's only working together that we're going to get answers. So I really am very grateful for your interest and your willingness. So thank you for that.
That's amazing. If folks on that note would like to reach out to Lupus Canada, if there is an interest in volunteering to donate their DNA to these studies, let us know, and then we will connect you with Dr. Hiraki.
Great, thank you.
I think there's one more question.
Yes, it says, if I'm a part of the lupus clinic patients, am I automatically in these donations?
No, not automatically, because you need to explicitly consent to provide your, not only your DNA, but for us to be able to link it to all of your clinical health history. So you can certainly speak with your rheumatologist to see if you can enroll in research, or again, like Leanne and Ariana kindly mentioned, you could contact them and we can try to figure out a way to get you enrolled.
Fantastic. Thank you so much. I think if there's no further questions, we will conclude our webinar. And again, thank you so much, Dr. Hiraki, for what a wonderful presentation. We've been looking forward to this one for a while. So thank you ever so much. Thank you, Ariana, for moderating this evening. And thank you to all our attendees. We hope you found it informative. And as we said, it will be available on our website within about 48 hours. And please reach out to Lupus Canada at info@lupuscanada.org should you wish to volunteer your DNA and be part of a study. Thank you, everyone.
Thank you. Good night.
Thank you. Have a good night.